The floor is yours.
Uh,
Take it away.
all right, fantastic. Thank you. That was really very generous introduction, and I'm just thrilled to be presenting to this community. And I really want to thank the organizers for inviting me, and also for putting this community together. I think it's just been such a fantastic experience. If any of you are interested in more details about the story that I'm going to tell you about today, you can scan the QR code, which is in the bottom right of this slide. And this slide is just a recent photo of some of the current and recently former members of our group. And I'm very fortunate to have collaborators both at UCLA and beyond, some of whom are listed on this slide, and their contributions to this particular project I'll mention as I go through. So our lab, as Axon mentioned, is really interested in the plasticity of the mammalian heart. And this is really important, particularly when we think of cardiovascular disease, which is the leading cause of morbidity and mortality worldwide. So you can see from this slide, there are a myriad of environmental factors that combine with genetics to control the basic function of the heart. And the key point I want to pose in the beginning is that the end stage of many forms of cardiovascular disease is something called heart failure. This is a syndrome that in the United States, for example, affects about 6 million Americans and has a 5-year mortality rate of about 50%, which is as bad or worse than most cancers. And just by one way of additional slide of introduction is to sort of think about what's going on in the heart. And there are many types of cells, all of which play important roles in normal physiology, as well as disease of the myocardium. But in our lab, we focus particularly on two types: the myocyte, this is the contractile cell, which is responsible for the beating of the heart. And I'm not going to talk too much about that today, but there's another subset of cells called fibroblasts, which exist across most organs in the body. And like in other organs in the heart, they're responsible for, in large part, the creation, the deposition, the maintenance, and the expansion of the extracellular matrix, as shown here in these slides, of a healthy heart and a diseased heart on the left and right, respectively. And it turns out that the biomechanical reprogramming of the heart in disease is a critical factor in determining the progression of these diseases. And so, again, as was mentioned, we started looking at this from the standpoint of epigenetics a couple of years ago. And what led us to the project I'm going to tell you about today is a proteomics experiment we did in a disease model. And I'm not going to go into any of the details, except to say that in the disease model, which is the green bar here, we looked at the ratio of linker to core histones. So H1 family histones to core nucleosomal histones. And we saw that that was decreased. And this was about 10 years ago. And we didn't know what to make of this, and we started to think in sort of crazy terms like, well, okay, is a 15% in the ratio of core to linker histones really significant? We thought, well, okay, if this was spread across the genome, then it could have direct effects on thousands of genes and maybe indirect effects on many others. And so now, fast forward about 10 years later, a recently departed postdoc in the lab, Dr. Shui Hu, who now runs her own lab at Tongji University in Shanghai, took on this question. And this is the conclusion, and then I'll walk you through the data. So as probably everyone on this call knows, linker histones, or the H1 family, is responsible for transitioning sort of beads on a string chromatin into higher order, more compact chromatin structures that then organize in the nucleus into chromosomal territories. And what we found is that the ratio of linker to core histones plays a key role in global chromatin compaction. This is not new, but it's a novel observation in the setting of cardiac fibrosis. But what is novel, and we think very interesting, is that this relationship between local and global chromatin compaction through H1 controls various biomechanical behaviors of these cells, including the deposition of extracellular matrix, cell migration and proliferation, the formation of these cells into contractile units as well as cell stiffness itself. So when we started this study, we wanted to know whether the H1 family plays a role in stress response, and particularly could H1 be involved in biomechanical behaviors. So these are some data from different cell types in the heart showing you the different members of the H1 family. The take-home message from this slide is that across multiple species, either the human or mouse, it's also true in other mammalian species, H1 is the isoform that's most enriched in cardiac fibroblasts as well as other cells. And if we look across different cell types in the heart, fibroblasts express the lion's share of the H1.0 isoform. If we look in single cell data, this is also true. So here, showing you just the individual H1 isoforms colored differentially, you can see that H1.0 is the most abundant across different populations of cardiac fibroblasts. At the transcript level, and when we look back at this paper that I mentioned earlier from a couple years ago, the other one here that I covered up was 1.2. H1.0 and 1.2 are the most abundant at the protein level as well. So it's preferentially expressed as it involved in myofibroblast activation. To do this, we took the myofibroblasts out of the heart and use them in primary culture, in which we stimulate them with a cytokine called transforming growth factor beta, or TGF beta. You're going to see me use this throughout the rest of the talk. What happens to these cells when you treat them with TGF beta is a very characteristic transformation in cellular morphology, which you can see here by alpha smooth muscle actin staining. They express smooth muscle actin, so they go from a quiescent state to a myofibroblast state where they can begin to contract, and I'll show you data on that. And then they also express various extracellular ligands and extracellular proteins, including periostin, which is shown here. So we wanted to ask the simple question, what happens if we modulate the level of H1.0 in this system? To do this, we took a sort of traditional pharmacological approach where we inhibit, or we deplete rather, H1.0 using siRNA prior to the administration of TGF beta. And I'm just basically showing you here that this works at both the transcript on the bottom as well as at the protein level to deplete H1. And it is sufficient to block the activation of periostin and alpha SMA. And this is just showing that data here quantified again with microscopic images. So what I'm showing you is that the expression of these traditional myofibroblast factors is dependent on the presence of H1.0. We also tested the other histone isoforms, H1 isoforms. We can get into this if folks are interested, and it seems to be specific to 1.0. So it's necessary, but is it sufficient? So it is. If we overexpress H1.0 in the absence of TGF beta, you can see that we recapitulate that myofibroblast phenotype, as shown here again by alpha SMA staining. So if it's necessary and sufficient for myofibroblast activation by these biochemical and histological markers, does histone H1.0 control the mechanical behaviors of the cell? To do this, we looked first at a contractile assay. So again, when we hit these cells with a cytokine, this is on the left here, a syncytium of cells on a gel, which they can then contract when exposed to TGF beta. And you can see that this TGF beta induced contraction is prevented by depletion of 1.0. That's on a group of cells. What if we look at the individual cell basis? Well, in collaboration with Jen Davis at UW, we did just that, and we see the same behavior there. This contraction phenotype is prevented by depletion of H1.0, and if we overexpress 1.0 in the absence of TGF beta, we can reproduce that phenomenon. This is also true for other phenotypes, including cell proliferation. So these cells will proliferate over time. We can prevent that by depleting H1.0. And another thing that these cells do, which is very important in vivo, is they will migrate to heal a wound. This is true for skin, it's true in the heart, it's true in basically all organs. And this TGF beta induced proliferation in mouse primary cardiac fibroblasts, here we've also done this in other cells, in other fibroblasts, is prevented by depletion of 1.0. And again, overexpression recapitulates it in the absence of TGF beta. So how does this work at the chromatin level to influence transcription? This is the question all of you are asking. when I show you this phenotype data. So there's a lot of really interesting things that are going on here. So if you look at the total H3K27 acetylation mark, either by Western blot, as shown here on the left, or by ChIP-seq, there are some very interesting findings. So there's a global increase in H3K27 acetylation. But if we look at the genes by ChIP-seq, where this reorganization in this enhancer and activating mark is going on, it's happening at very specific locations, okay? So if we look at the genes that are upregulated by TGF-beta, but whose upregulation is prevented by depleting H1, we see that the enrichment in those marks that occurs with upregulation is also prevented. The antithetical situation is also true. So if we have downregulated genes, unsurprisingly, in the TGF-beta condition, these tend to be areas where we don't see H3K27 acetylation. And it's not as though all of a sudden we have an enhancer forming when we deplete H1, but you can see that the relative depletion of K27 in those regions is lost when we deplete H1, suggesting that one mechanism by which this is happening is by reprogramming this enhancer mark at specific locations around the genome. And we wanted to tease this out a little bit further, and the way we did this, many of you are familiar with the Bet-bromodomain family of proteins. So a characteristic member of that family is BRD4, which is known to be operative in cardiac fibroblasts. And these proteins bind to acetylated lysines and coordinate transcription machinery recruitment. And so we asked the question, what goes on with BRD4 localization, in this case to 3 specific genes that we know are involved in fibroblast activation. 2 of these I've been talking to you about so far, alpha SMA and periostin. Another one is called SIRTAD4. And you can see in all 3 of these conditions, we see recruitment of BRD4 to these loci following TGF-beta treatment. But if we deplete H1 in advance, it's sufficient to block this phenomenon. This is an MNase assay that we use to examine this. Usually when I explain this to cardiovascular audiences, I have to explain this assay in detail. Here, of course, I don't. As you know, the MNase will digest less when there are higher order chromatin structures. And so if we overexpress H1, we can see this is an electrophoresed set of DNA fragments here, that the chromatin is protected against MNase digestion. And if we deplete H1, we see the antithetical situation. This isn't anything new. This is what you would expect. But what we found was very interesting is that TGF-beta tends to condense chromatin. And that chromatin condensation is prevented if we deplete histone H1. Another way we looked at this is global chromatin condensation across the entire nucleus, shown here by DAPI staining and quantified here on the right in a chromatin condensation parameter. And this is just a control to show you that if we treat them with hypotonic or hypertonic solution, respectively, the chromatin will decondense or condense. And we see the same behavior when we modulate the abundance of H1. So depleting H1 causes nucleus scale chromatin decondensation, and overexpressing H1 has the opposite effect. So then in closing, we wanted to ask the question, what if chromatin itself could be behaving as a mechanosensor? And to do this, one of the things that we did was we used atomic force microscopy to examine the relative stiffness of different regions of the cell after depleting or overexpressing H1 before the cell itself changes its stiffness. Okay, so this is quantified here in Young's modulus. So this is basically a measurement of cellular rigidity or stiffness. So if we look in the H1 depletion scenario, the nucleus becomes less stiff. Okay, and we see a modest but non-significant effect on the cytoplasm in the same scenario. And when we overexpress H1, we see the opposite behavior. And an important observation here is that the cells have not yet adopted this myofibroblast phenotype. Okay, so they've not stiffened, but the nucleus itself is stiffening in response to changing the abundance of H1. Another way that we did this was to look at the actual deformability of the whole cell, which it turns out in these cells is modulated by nuclear stiffness. And we did this in collaboration with Amy Rowat here at UCLA. And what Dr. Rowat does is she fabricates these porous membranes through which one can basically try to push cells with some air pressure. And the more deformable the cell over a fixed period of time and fixed pressure, the more cells will go through the membrane. The less deformable cell, more of them will be retained up top, right? And so here we're measuring retention, and you can see that if we deplete H1, the global nuclear deformability is much greater, okay? There are fewer cells that are retained up top. More of the cells are getting pushed through in a fixed period of time, indicating that the nucleus is more deformable. And this is very interesting because in conditions like cancer, when cells have to extravasate through tissue in metastasis, the nucleus is known to become more deformable. In fibrosis, in healing of various injuries, including in the heart, this is also the case. It turns out many times the things that we want to inhibit in cancer, we want to activate in cardiomyocytes to promote cell death instead of inhibit it. But in fibroblasts, the behaviors we're seeing here are probably more like some of the behaviors in cancer. And then again, the opposite scenario is true. If we overexpress H1, we see more cells retained, indicating that the cells are less deformable and the nuclei are stiffer. So lastly, are the mechanisms that I've been talking about here, are they operative in vivo? And could we use what we've learned about the basic relationship between histone stoichiometry to have a different understanding for how fibroblasts are activated in the setting of injury? And I think a broader question is, what is the role for direct coupling of chromatin structure and gene expression to cellular stiffness and tissue stiffness? So to examine this, we used a mouse model of fibrosis, this condition that I told you about in the beginning today. So we used two different mouse strains. Again, we can go into detail with this if anyone's interested in the Q&A, but we treated these mice with a beta-adrenergic agonist, which is called isoproterenol, for several weeks, and then we injected them in the tail vein with siRNAs against histone H1.0. You can see we got good, knockdown in vivo. And the level of H1, very interestingly, and in a sort of nice reality check, corresponded with a positive relationship to the level of periostin. So you recall periostin is the stress-activated marker that we've been using. So this all suggested to us that we were reproducing in vivo this behavior that we had been seeing in isolated cells. This is some physiological data here. I just want to call your attention to two things down here. Again, these are the two different mouse strains. And if we look at the green ones versus the black, we see that in the isoproterenol treated animals, the hearts are getting bigger, okay? Cardiac hypertrophy is a key feature of heart disease that's initially compensatory, but ultimately causes a worsening of the disease. If we look over here on the right, again, the labeling is the same. This is a measurement of the degree to which the ventricle relaxes, okay? So the ventricle is the main pumping chamber of the heart. If it is fibrotic, if it's stiffer, then it's not going to relax as well, okay? And that's what we see here, is a stiffer ventricle in the setting of isoproterenol, and this is prevented by depleting H1. And then lastly, this key readout that we have of the progression of cardiac disease, and something that we know we need to address if we want to impact the outcome in this disease, is fibrosis. And so this is histology here. This is a cross-section of the left ventricular chamber, and the blue staining here is the fibrotic tissue. And you can appreciate that after isoproterenol, this fibrosis is expanded, okay? But that fibrotic expansion is significantly blunted by in vivo depletion of H1, suggesting to us that these mechanisms I talked about in the beginning may be operative in vivo, and that these biomechanical behaviors, the sensing of tension and stress by the cell and by the nucleus, may communicate in both directions to control gene expression and to link chromatin structure with these biomechanical properties of the cell, and could be a possible new way of looking at biomechanical remodeling and fibrosis and diseases such as heart failure. And with that, I'll stop. I think I mentioned others while we went through the talk. I'd be happy to take any questions.
Awesome. Thank you so much, Tom. As always, if you have any questions, you can raise your hands, or you can enter something into the Q&A box. Ben, you've already got your hand up. If you want to go ahead and ask a question.
Yes, thank you, Tom. That was fantastic. Really interesting work. I've got many questions, but I'll start with just one. And I was wondering, with all the work you've done has been focusing on H1.0. You've got really nice data with that. And I was wondering if you have any sense of it, is this a matter of just dosage of H1 proteins, or is there something specific about 0.0 versus all the others?
Yeah, it's a great question. I think it's, as far as we know, it's a dosing issue. So the 1.0 is the most abundantly expressed isoform. That's true in cardiac fibroblasts as well as fibroblasts from multiple other organs. It's also true in myocytes. I showed very briefly the protein data. So the protein data is much more complex and very interesting. So 1.2 actually is more abundantly expressed in the heart at the protein level. You know, and I think a reason why this field of linker histones, at least in terms of understanding their role in phenotype, has been so challenging is, you know, there's work done by Art Skolchi and others starting over 30 years ago, where they showed very nicely that if you deplete any one of the isoforms, the cell is very good at upregulating others. So he had to knock out 3 isoforms before he got a phenotype. And he's done some other really nice work that frankly inspired this project to get us thinking about the ratio of linker to core histones. And I think why what we're doing works is because it's a transient depletion. So we know that the time frame that we're operating under, the other isoforms have not yet been upregulated. And so this is sort of a long-winded answer to your question, is I think the cell is very good at exchanging out other linker histones. We think there could be something interesting about 1.0, but we think that probably what we're sort of getting at here is a more fundamental relationship between chromatin condensation, nuclear stiffness, and cellular stiffness.
That's, yeah, no, it's really interesting. And then one maybe quick follow-up, which is, have you, considering that, you know, if it is dosage, have you looked at how, like how this upregulation is happening? Like how is H1.0 expression controlled versus the other ones?
It's a great question. Short answer, we have not looked at it.
Okay, thank you.
I also have a bunch of questions, but I'll go ahead and ask this one in the Q&A first. Alan Underhill asks if you see a relationship with heterochromatin marks like K9 trimethylation that are also known to regulate the rigidity and responsiveness of chromatin.
Yeah, it's something we're very interested in. So I didn't have time to go through the data today, but we see changes in multiple chromatin marks. Either by Western blot, by ChIP-seq, or by whole cell immunofluorescence. So you mentioned H3K9 trimethylation. That one in particular, we see a decrease in that mark when we deplete H1. And we also see that there are puncta of that mark that form and that change their relative level of condensation when we modulate the level of H1, when we deplete H1. We don't know what that means, but we think it's probably related to some higher order chromatin organization. It's a really interesting question that we want to try to answer.
Awesome. So then, like at the molecular level, like whenever I think of the linker histones, I think of their similarity to like the winged helix domain and pioneer factors like the foxes. Do you think that this sort of um, role for them in being like a barrier in the genome, do you think it has to do with them just being a barrier to pioneer factors entering into these sites to activate them in response to these stresses?
It's a fantastic question, since we're not dealing with a developmental question here, and I guess just as a sort of side comment on cardiac fibroblasts, it's a heterogeneous population of cells. So we don't know, like, okay, these are definitely the transcription factors that we would need to look at from a pioneer sort of model to ask that question. There are, I showed a lot of data with TGF-beta induced changes. This is this process is also maintained in response to other stimuli, but TGF-beta, for example, is known to couple to SMAD transcription factors. And this is something that we're very interested in. And whether H1 itself is acting like a physical barrier on chromatin or condensing something to make it inaccessible, this is a very attractive explanation. We think that could be part of the reason for why we're seeing what we see with BRD4. But it's really unclear.
And then do you think that there are other chromatin operations or functions in the chromatin that are required to evict or remove H1 as the chromatin is transitioning from this more compact state? Or is it more labile? Like, does it need like an ATPase to come in and remove it?
Yeah, we don't have any, I can't really comment on that. We don't know the direct answer to your question, but there's actually something else very interesting in what you're asking is, I'm showing you all of this stuff with, we're depleting this using siRNA. We've also done it with shRNA. But the question really is, what role does this play in sort of normal physiology? This is why we thought it was so interesting that we saw these global chromatin changes in response to TGF-beta that have nothing to do with modulating the level of H1. And so I think that we know that H1 expression is dynamic in response to various disease states or stressors, but we think that by modulating its stoichiometry, we're sort of uncovering aspects of this behavior that may involve linker histones, but also involve other proteins. We and others have looked at similar sorts of questions in response to modulating levels of CTCF, which also is, as I'm sure everyone knows, responsible for higher order chromatin structure. And so, a lot of these disease states, a lot of these pathologic activation of cells can be modulated by chemical inhibitors that modulate HDACs or modulate BET bromodomain proteins. And a lot of times we think of these things as very like locus specific, and it's got to be regulating this gene. And I think there is some of that, but I think there's also an effect to, regulate the basal plasticity of chromatin that allows the cell to adopt different fates.
Yeah, that's super fascinating to think about. And so I guess as we're coming up on 1 o'clock here, doesn't look like there's any more hands up in the Q&A. So I'd like to once again thank Tom and Remy for 2 just outstanding talks, and anyone else in the audience, I'm sure if you have questions, you can pop into the Fragile Nucleosome Discord and interact with our community there.